from Joanne Stubbs' lab in MIT, and then after that he went on postdoc with a NIH postdoc fellowship with Chris Wash, and then he started Penn State faculty job, and he's a professor in chemistry and biochemistry and molecular biology department, Penn State. He teamed up with Dr. Carson Kratz and has over 100 publications, including some of the recent Science and Nature papers, and he's an AAS fellow, and he received many awards, including but not limited to Penn State Faculty Scholar Medal for Outstanding Achievement, Society of Biological Inorganic Chemistry, Early Career Awards, etc. And I would like to point out he also hosts this bioinorganic workshop in Penn State University with Dr. Carson Kratz, and I personally benefit a lot from that workshop. If you were to study the metalloenzymes and study the deep biochemistry of the metalloenzymes, I highly recommend this seminar, workshop. And without further ado, Mark.
Thanks, Ray, and I should thank Aaron Insan for inviting me and then disappearing, not being here. So it's great to come, and I look forward to talking to people later today. What I'm going to tell you about is our efforts to understand the second of two steps in a two-step enzyme pathway by which people are developing bioprocesses to make actual diesel fuel, not biodiesel as in fatty acid esters, but actual diesel fuel in bacteria. And the enzyme, we coined the name aldehyde deformylating oxygenase. As you'll see, it was originally called something else. And so the story began in around 2009, 2010, when a biotech startup company out here, LS9, reported a two-step enzymatic pathway converting saturated or monounsaturated fatty acids of chain length C14, C16, or C18 from linked to acyl carrier protein to the corresponding saturated or monounsaturated fatty aldehyde, and then ultimately to the alkane or alkene. And so the chain length here, as occurs in the cyanobacteria where the pathway is found, is about the same distribution as in the diesel fuel that we buy from the pumps. And what the authors of this study at LS9 pointed out was that the genes and enzymes they discovered could be leveraged for a simple direct conversion of a renewable raw material, that being abundant fatty acids, into a drop-in hydrocarbon fuel. And so this idea got generated a lot of excitement. What you can imagine is a pathway driven only by light, with the feedstock being only CO2, whereby the light reactions of photosynthesis make reducing equivalents to reductively fixed CO2 into precursors to and ultimately to the fatty acids, which can then be siphoned off by this very simple two-reaction pathway to make diesel alkanes. And so a number of companies have sought to cash in on this opportunity, LS9 being one of them. And their idea was to import the pathway along with any accessory factors that might make it efficient into genetically tractable hosts like E. coli and other good laboratory host bacteria. And we got involved with LS9 because they needed to understand the pathway to know what else they needed to import into these heterologous strains in order to make the pathway efficient. And here you can see that as of 2012, they had purchased and retrofitted this production facility. They call it a demonstration scale facility, and here are some presumably potential investors and LS9 scientists and administrators examining these cultures from which the diesel has been made and is, as you can see, phase separating away from the culture. And that's one of the really attractive aspects of this potential bioprocess, is that the separation is almost trivial. It amounts to a centrifugation, and LS9 applied one very low-cost purification step that gave them diesel fuel that's purer than what we currently buy at the pumps. And so that was LS9's vision. They've since been bought. Joule Unlimited in Cambridge, who I see kind of as their competitors, but they don't, it's not clear to me the relationship between the two companies. Their vision is to use the actual cyanobacterial hosts, and which, of course, cyanobacteria are photosynthetic. So here they have a demonstration scale plant with this array of fluidic solar panels to allow the cultures to be exposed to sunlight so that they can do photosynthesis. And they're infused with industrially sourced CO2 from, for example, coal-fired power plants. And then there's a separation facility, and here you can see some Joule scientists examining the cultures. So I think in addition to the activity in biotech and supported by the energy industry, and I'm willing to bet almost everyone in this audience knows more about that than I do, I think that the flurry of publications in technical journals in which scientists, academics in particular, have sought to engineer this system for more design properties speaks to the excitement that it's generated. And so we've seen, just in the last 2 years, fusion of the aldehyde deformylating oxygenase enzyme to catalase to combat inhibition, and I'll talk a little bit about this inhibition by hydrogen peroxide. We've seen directed modification of the ADO to make variants that favor short-chain alkane production, and down to including propane. We've seen modification to the coli fatty acid metabolism to tailor the pool to get direct products. We've seen the engineering of alkane efflux pumps in yeast to combat the toxicity that one gets when one makes high concentrations of these alkanes in living organisms, such as bacteria and yeast. We've seen modifications to the pathway for different products like fatty alcohols, and most recently seen a fusion of the ADO enzyme to this ferredoxin reductase system that supplies it with the electrons it needs to be efficient, and I'll talk a good deal about that. We're not doing any of this bioengineering. What we're interested in is fundamentally understanding the enzyme, in particular the second enzyme in the pathway, and how it works, it cooperates with the first enzyme, how it interacts with the first enzyme. And we believe that fundamental understanding will help people who are doing the bioengineering to do it better and make the system more efficient. And so I will, though, talk about some interesting observations with regard to hydrogen peroxide inhibition and this notion of the challenge that the enzyme faces in terms of delivery of electrons from these accessory factors. So aside from the biotechnological hype surrounding the system, we were interested in the actual chemistry, in particular this second reaction, which was originally described by LS9 as the enzyme was called aldehyde decarbonylase, and the C1-derived co-product here of the aldehyde group was proposed to be carbon monoxide. And in reading their paper, we were immediately struck by a puzzle. It turns out that there wasn't a lot known about enzymes that could do this sort of decarbonylation reaction. There had been some sketchy reports, but none of the enzymes proposed to do that reaction had been characterized in any depth, so they weren't understood. And when one looks to the chemical literature for precedent about aldehyde decarbonylation catalysts, one finds almost exclusively organometallic type catalysts. So one particularly well described catalyst is this rhodium-1 carbonyl bisphosphine complex that can undergo an oxidative addition of the aldehyde, which displaces carbon monoxide, one of the products, and makes this acyl hydrido bisphosphine rhodium-3 complex. There's then a CO extrusion step which splits the alkyl group from the CO to make this alkyl hydrido carbonyl bisphosphine rhodium-3. Then there's a reductive elimination where the alkyl group gets together with the hydride to make the alkane and regenerate the starting catalyst. So what you can see is this reaction is redox neutral. There's no input of or extraction of electrons during the cycle. But we didn't expect to find rhodium in the enzyme that does such a reaction in cyanobacteria. And in fact, LS9 was able to show what the cofactor was likely to be. And they were able to report the structure of the second enzyme, which they called aldehyde decarbonylase, because the Joint Centers for Structural Genomics had solved an enzyme of this type without knowing its function. So LS9's work allowed a function to be associated with this known structure, and the structure is shown here. LS9 discussed it and compared it to an enzyme that protein that I worked on as a graduate student with Joanne Stubbe, and this is the R2 subunit or beta subunit of ribonucleotide reductase from E. coli. So these proteins are both members of what we call the ferritin-like diiron carboxylate family of oxidases and oxygenases. And you can see that most of the members of this family, including the ribonucleotide reductase subunit and the so-called aldehyde decarbonylase, have diiron centers at their active site. So they have diiron cofactors. And what enzymes in this family do are almost exclusively oxidation reactions. So it's known from work in my group and John Lipscomb and Steve Lippard and Brian Fox and John Shanklin, Vincent Wynn and others that the way these enzymes work is that oxygen adds to the fully reduced diferrous form of the cofactor to make, in general, a bridged peroxodiiron 3 intermediate. Now these may have different structures in the different systems, and that structural differences may be important in dictating the outcomes of the reactions. But we formulate this as this most general structure here. And this peroxodiiron 3 intermediate can undergo a redox neutral OO bond cleavage in, for example, soluble methane monooxygenase to generate this bis-mu oxodiiron 4 intermediate, which has been called Q, compound Q in methane monooxygenase. And it can hydroxylate alkanes, in particular methane to methanol. It's not known, but it's reasonable to presume that a similar intermediate is responsible for dehydrogenation of fatty acids in plant fatty acyl ACP desaturase enzymes to introduce double bonds between previously saturated carbons. We showed that the arylamine oxygenases in biosynthetic pathways to antibiotics that contain arylamines are produced by the reaction of such an intermediate directly with an amine group on the arylamine to make the aryl hydroxylamine, and that this intermediate is then further processed by the same peroxodiiron 3 complex fully to the aryl nitro complex product. It's likely that a similar intermediate reacts directly with an olefin in alkene monooxygenase to make an epoxide. And my work as a graduate student with Joanne Stubbe, and then later in my own group, worked out the reaction by which the tyrosyl radical is introduced into the R2 subunit of ribonucleotide reductase. It's the enzyme's essential cofactor, and it happens when peroxodiiron 3 intermediate is reduced by 1 electron to make this iron 3, iron 4 complex, which then oxidizes the tyrosine by 1 electron to the stable radical. So what these reactions have in common is they're all oxidation reactions, right? They're all 2 electron oxidations along the entire span of that slide. But the reaction proposed by LS9 for this aldehyde decarbonylase is redox neutral. What's more is LS9 showed that in order for this enzyme to have activity in vitro, they had to include a reducing system. And what they used was actually a ferredoxin reductase from spinach, and its chemical reductant NADPH. They had that in the refrigerator, and Matthew Rudd just tried it, and it worked. And they needed this reducing system for any in vitro activity. And that is also common to this family of enzymes. And the reason they need a reducing system is at the end of a single turnover, the cofactor has been converted to not the single turnover, a half turnover. The cofactor has been converted from its reduced diiron 2 state to its oxidized diiron 3 state, and that state's not reactive with oxygen. In order to undergo a second turnover, the reducing system has to return the cofactor back to the O2 reactive diiron 2 state. And so this so-called aldehyde decarbonylase had both structural and functional hallmarks of a ferritin-like diiron carboxylate oxidative enzyme, but the reaction they proposed was, in fact, redox neutral. So it's not clear that LS9 appreciated the conundrum that their proposal created, but it's absolutely clear that their competitors at Joule did, because in Joule's patent application, which was filed after LS9, they actually proposed that the reaction results in conversion of the C1 carbon into carbon dioxide, not carbon monoxide. Now this would be a 2-electron oxidation, which would account for both the structure of the enzyme and its membership in this family, and the fact that it needs a reducing system, because this reaction would need 2 electrons and 2 protons along with molecular oxygen to balance it. So we kind of liked that idea, but when we looked into it, Doug Worry and Squire Booker's group at Penn State and Ning Li and Carsten Krebs and my joint group found out that neither patent application was correct. The actual C1-derived coproduct is in fact formate. Okay? So now if you look at how formate gets produced from an aldehyde, this looks like it could be a weird kind of hydrolytic reaction. If you imagine, as a thought experiment, water attacking the carbonyl, you can have a carbanion leaving group which you would protonate, and you get the products then. You get formate and you get the alkane. Now that's a weird mechanism, but this is exactly the reaction that was later proposed a couple of months after we published on this system by the Marsh group at the University of Michigan. And so we became embroiled in a controversy that stretched for about 18 months about the nature of this reaction, but I think it's been resolved. And what we showed is, what Ning and Doug showed, is that the reaction is actually a cryptically-redox oxygenation reaction. And I'll explain what that means. Oxygen, one equivalent of oxygen is required in every turnover, but it doesn't do an overt oxidation of the substrate. The products look like hydrolytic products, but the oxygen is reduced by 4 electrons from the reducing system. And somehow that the coupling to oxygen reduction gives the chemical punch needed to do this unusual carbon-carbon bond cleavage reaction. And what's more, and what's important, is 1 atom from O2 is actually incorporated into the formate product. So that's not a hydrolytic reaction. We call it a cryptically-redox oxygenation reaction. And so as I said, there was a controversy even after we published what we considered pretty conclusive evidence in this paper. And so I'll show you just the experiment that I think basically ended the controversy and led the Marsh group to publish 2 corrections, essentially acknowledging the role of oxygen and the oxygenative nature of the reaction. And so what this experiment was examining the fate of, or the identity, the origin of the oxygens in the formate product in reactions run in O18 water. Okay, so we're labeling the solvent here. And what you have to know, as a chemist you might know this already, is that when aldehydes are put into water, the oxygen of the carbonyl undergoes fairly facile exchange with the solvent. So if we put this aldehyde into O18 water, the carbonyl is going to be exchanged. Well, the Marsh group said that took many, many hours. But when Wei-Chen Chung and our group looked at it, and we used decanal or octanal, either one, as the substrate to balance efficiency of turnover with solubility of the substrate, because the longer chain substrates are not very soluble. So Wei-Chen looked into this, and what he found was the exchange of oxygen with solvent occurs with a half-life of about a minute. So if we just wait 5 minutes after mixing the aldehyde into the O18 water, we now have an aldehyde substrate that is fully equilibrated in terms of the oxygen with the solvent. Okay? And then the experiment becomes just assessing where does the second oxygen come from. We know one comes during exchange from solvent. Does the other one come from solvent in a hydrolytic reaction or from molecular oxygen in this cryptically-redox oxygenation process? So Ning Li did this experiment tracing the oxygen. And what he did was he ran LCMS experiments, and here he shows single ion monitoring chromatograms for M over Z equals 46, 48, and 50, corresponding to C13 labeled formate. We always use C13 to separate it from environmental formate, containing 0 O18s, 1 O18, or 2 O18 atoms in the product. And he ran two reactions in a control, all in 85% O18 water. Okay. In the control, in the absence of oxygen, he saw no product at any of these M over Z values, and that was consistent with our original publication saying that oxygen was required for this reaction. The Marsh group had claimed that oxygen wasn't required. So again, we showed that there no oxygen, no product. When Ning ran the reaction in this 85% O18 water, but natural abundance O16 gas, what he saw was a 15% formate with M over Z 46 and 85% with M over Z equals 48, precisely the isotopic composition of the solvent. What he notably didn't see was any peak whatsoever for formate containing 2 atoms of O18, the first from solvent in the exchange, the second from a hydrolytic cleavage event as proposed by the Marsh group. And so there was absolutely no formate containing 2 atoms of oxygen from the solvent. And just to close the loop, Ning then ran the third reaction in O18 solvent and O18 gas. And when he did that, the peaks from the formate product shifted back 2 mass units, 15% M over Z equals 48, 85% M over Z equals 50. So what this experiment conclusively shows is that there's precisely 1 atom of oxygen from the solvent that comes in the exchange process, and precisely 1 that comes from molecular oxygen during the cleavage of that carbon-1, carbon-2 bond. Okay, so we can get that. And so upon viewing these at a Gordon conference a short time before this paper came out, the Marsh group published 2 corrections, and we could all look ahead to trying to understand how this reaction happened. So in that Jack's paper in 2011, we had proposed a mechanism for the reaction. And this was based on our understanding of this family of enzymes. And in the mechanism, what we proposed is that the enzyme binds the aldehyde to one of the irons of the cofactor. And then oxygen then adds to the diferrous form to make what we draw here sort of an asymmetric mu A to 2 peroxodiiron 3 intermediate. So this is that canonical peroxodiiron 3 that I showed on the slide with all those different reactions. But uniquely in the ADO reaction, and it was at this point that we renamed the enzyme from aldehyde decarbonylase to aldehyde deformylating oxygenase. There's a really interesting story about that I can tell at lunch if anybody's interested. But uniquely in this reaction, one of the oxygens of the peroxide acts as a nucleophile to attack the carbonyl of the bound substrate to make this diiron 3 peroxyhemiacetal intermediate. The next proposed step is a reductive cleavage of the OO bond here. So the reducing system now dumps in an electron, and we get this gem diallyl radical and a mu-oxodiiron 3 cofactor form. This radical undergoes a beta scission to make the formate product bound to one of the irons and the alkyl radical. And then the alkyl radical acquires a hydrogen atom, either from maybe a solvent bound to the cofactor or from a dedicated hydrogen atom donor, an amino acid like a tyrosine or a cysteine in the enzyme active site. And the enzyme then acquires an electron from the reducing system and a proton to give this product form of the enzyme with formate and alkene bound. Dissociation of the products, rebinding of the substrate, the aldehyde, and delivery of 2 more electrons and 2 protons by the reducing system regenerates the starting complex. And we don't know what order those events would happen. We haven't studied them yet. But we get back here to the reactant form. Okay, what did I want? Well, so you should note that in the overall stoichiometry there are 4 electrons. 2 come in reducing this product form back to the O2 reactive reduced reactant form, just as occurs in the other members of this family. But 2 more occur uniquely in flight during the mechanism with reactive intermediates formed to cleave the OO bond to the peroxide, and then subsequently, once the alkyl radical has been quenched, or possibly to quench the alkyl radical, there are 2 more electrons delivered, total of 4. So oxygen is fully reduced, the substrate is not overtly oxidized, and we can account for all the products according to this mechanism. I should say that this mechanism was developed really without any good structure. We didn't have a structure of the reduced cofactor with the aldehyde bound, but as of 2 weeks ago, the Marsh group actually published such a structure. Now, the substrate is non-native. It's got this polyethylene glycol appendage here on the fatty aldehyde, the linear fatty aldehyde, to make it soluble. So that was a clever trick to be able to get the structure. And it's putatively in the reduced state. And what you can see is the aldehyde does indeed bind to 1 iron of the cofactor and poises the other one, maybe to activate oxygen to add into the carbonyl group. But it turns out that the original structure had a fatty acid bound, and that structure is very different from what Marsh sees in the structure of the reduced form with the aldehyde bound. So this structure will be important in going forward to assess computationally to interpret our spectroscopic data on intermediates and to computationally interrogate the reaction coordinate. At the moment, we couldn't do anything with it because it only appeared 2 weeks ago, and the coordinates haven't been released. So I just want to point out that that structure should be available sometime soon. So we wanted to test this mechanism even in the absence of a good structure, and we sought first of all to see if we could get evidence for the first 2 steps, the addition of oxygen to make first a peroxide intermediate and then some sort of peroxyhemiacetal. So the first thing we had to be able to do was to get the reduced form of the enzyme, and both Ning and Maria could do it. Ning did it first, and Maria kind of perfected it, either by isolating the enzyme from overproducing E. coli anaerobically, or by treating the aerobically isolated form, which comes out oxidized, with the reductant dithionite. Either way works. And so here Maria shows you what the Mossbauer spectrum of the aerobically isolated form looks like. It's a classic antiferromagnetically coupled diiron 3 cofactor. This is what it looks like in high field. That's not really important. The point is, anaerobically isolated, you can see that the peaks move here, and what we get is a diferrous form that has only about a 6% contaminant of the oxidized diferric form. And so that's the form Maria preferred to use, the anaerobically isolated form. Okay? And so it's 94% in the reduced cofactor state and 6% oxidized. So Maria then put that reduced protein in a stop flow and mixed it against oxygen, and what she saw was really boring. She saw a very slow monotonic growth of the peak at 360 nanometers, which is classic for a mu-oxo diiron 3 cofactor. So what we're seeing is very slow, unproductive oxidation of the cofactor in the absence of the substrate. However, if she included the substrate, or when she included the substrate, in with the reduced ADO, now in this case 10 millimolar decanal was used, she saw something very different, and Ming before her had seen the same thing. Now there's a much more rapid oxidation with formation of some transient absorption centered at 450 nanometers. So this goes up and down, and the whole process is much faster. So we say that it's triggered by the substrate. Substrate triggering is common in iron oxygenases, and it's seen here in aldehyde deformylating oxygenase. And here's our first MAC to PC problem right there. So that's pretty good. Here's the second one. Darn it. So what Maria and Ning did then was to look into this substrate triggering of the reaction. They varied the chain length, because if the natural substrates are C14, C16, and C18, one might expect them to be best at triggering the reaction. And what they found out is with hexanal, it doesn't trigger as well. It's a little bit slower. Peptinal becomes a little faster. By the time you get to octanal, nonanal, and decanal, they're pretty close to being on top of each other. And so there is a dependence on chain length, but it turns out we don't need the full chain length, and that was lucky because the C14, 16, and 18 substrates are really insoluble and would complicate our Mossbauer experiments that require high concentrations. So what Maria did then was to examine decay of this intermediate after it forms and do the kinetics. And interestingly, what she found is that when she used the bright white light not needed for the PDA detector, the photodiode array detector, she saw this intermediate decay much faster than when she used monochromatic light and a photomultiplier tube detector. We see this all the time in reactive iron intermediates. They're photosensitive. And so this is photolytic, light-promoted, non-productive decay of the intermediate. And so we don't do any of the experiments with that bright white light. We do them with the photomultiplier tube. So the intermediate's light-sensitive. It had a mean lifetime of 7 minutes. So we're claiming this is a reactive intermediate, but it lasts for 7 minutes. So that's really slow. Forms after addition of aldehyde and oxygen, and it has a maximum absorption that you can't see of 450 nanometers. So this suggested to us, based on our experience on precedent, that this is a peroxodiiron 3 intermediate. So we asked the question, is it this thing or this thing? Because they're both peroxides with a diferric cofactor. Either one is a viable structural formulation. And, all right. So we sought additional spectroscopic evidence in the form of Mossbauer spectroscopy. So Maria now mixed the reduced enzyme with the substrate in syringe A and shot that against oxygen in syringe B, and now quenched the reaction by rapid freezing after 32 seconds. So she allows the intermediate to accumulate and then traps it by freezing. And here is the Mossbauer spectrum that she gets in the middle here. She can recognize a contaminant of unreacted ferrous starting material and the 6% contaminant of the oxidized protein that was present in the starting material, and she can subtract those away, and she gets the spectrum of this new intermediate state, which she can analyze as 2 superimposed quadrupole doublets, both with parameters indicative of ferric ions. So here are the parameters down here. Take my word for it, these isomer shifts tell you it's ferric, but the intermediate is markedly site differentiated. It has a quadrupole splitting parameter, delta EQ, that's much greater for one site than the other. So it suggests that it's asymmetric in some way. The 2 sites are resolved. Okay? Maria herself did the high field Mossbauer, and that gives a diamagnetic ground state. And so we can, at that time, we could accumulate it to 80%. We can now get over 90% of the intermediate. It has pronounced site differentiation, meaning it's not symmetric. Diamagnetic ground state and decays very slowly in the absence of a reductant. So now we ask the question, is it more likely to be something like this or something like this? And one suggestive bit of evidence is that we examined other alkyl compounds terminated by other functional groups. So the aldehyde basically gives you this intermediate, which forms and decays. But if we look at nonanal, that works too. This is decanal. Nonanal works. Nonanol, the alcohol, doesn't give you anything. It looks as though you didn't add any compound. So you could say it doesn't bind, but I can tell you later it actually does bind, and it triggers the reaction with hydrogen peroxide, but it doesn't trigger the reaction with oxygen. So what that says to us is the carbonyl is essential to forming whatever this intermediate is. It just doesn't develop if you don't have the carbonyl. If you have the alcohol, it doesn't work. So we like to believe that it's this peroxyhemiacetal state, and we like that in particular because of the site differentiation in the Mossbauer, because this gives a really easy way to say that this iron should be very different from that iron. This, with an aldehyde, it's not a very strong ligand. This could be more symmetrical. But once you make this sort of gem dialate type thing, or peroxyhemiacetal, that should site differentiate the cluster. So we think the intermediate is this guy. And the next question is, how can it be so stable? Well, it's stable because the next step is reduction, and we don't include a reductant. So what happens if you make that intermediate, and mix it with the reductant. The reductant we used at first was one reported by the Marsh group to be a very good chemical reductant. It's the methoxyphenazinium methyl sulfate. The reduced form of that, and the structure is shown here. So what Maria and Ning did in these experiments is to mix the reduced enzyme with its substrate against oxygen, accumulate the intermediate to its maximum extent, and then mix it with the reductant to see what happens. Okay, so in the control, Maria mixes, she leaves out the enzyme, reduces, does a dummy mix of nothing with O2 saturated buffer and mixes that with the reductant, and oxygen is able to oxidize this reduced phenazine, giving an absorbance of 388 nanometers, only very slowly. Okay, if she uses enzyme as isolated, doesn't make the peroxide, just mixes the dipheric form with the substrate, and then mixes that against the reduced phenazine, again, very slow oxidation, almost nothing happens. But if she makes the peroxide intermediate and then mixes that with the phenazine, you can see there's a couple of log units separation in time here. So this intermediate reacts very quickly with a half-life of about a couple hundred milliseconds with the reduced phenazine. Okay? So the rate constant here that she pulls out was about 5 per second. So the question is it productive? Do we get formate? And so they did the experiment where they made the intermediate, reduced it with varying amounts of the reduced phenazinium, and what they see is if you add no reductant, decay of that intermediate in the absence of reductant leads to very little formate production, 0.1 equivalent. But with increasing amounts of the reduced phenazine, see increasing production of the formate product. Now the slope here is only 0.5. Theoretical is 1. So it's only half of the value that you should see. It tells us the intermediate is chemically competent to make the product, but we have to understand why only half the decay events are productive. We think we have an explanation. So phenazine is a 2-electron, potentially a 2-electron reductant, and the trick here for this enzyme, the reducing system needs to deliver 1 electron and then allow time for this intermediate to fragment before the second electron is needed. What we suggest is that the potential 2-electron donor can donate 1 electron, and then in partition with the fragmentation, we can get donation of a second electron, which deactivates it. You need that radical to do the fragmentation. So we think this is an uncontrolled delivery of a second electron by this artificial chemical reductant. And we theorized that using the natural ferredoxin reductant, the biological reductant, might correct that problem. So then we said, well, if we get formate, do we get an alkyl radical? About the time we were, while we were doing these experiments, the Marsh group performed a really clever experiment in which they incorporated a cyclopropane radical trap into a fatty aldehyde. And what they showed is catalysis by ADO, which presumably would make a radical here. That methylene cyclopropyl radical ring opens rapidly, and they saw the ring open product. They also saw, in some events, covalent attachment of the original cyclopropyl fatty aldehyde to the enzyme with inactivation. And so they provided pretty good evidence for a radical mechanism in the form of the ring opening and the covalent inactivation of the enzyme. We were looking for direct evidence for a radical, so we did these double mixing experiments, making the peroxide, mixing with the phenazine, and Maria freeze-quenched now at 10 milliseconds, 40 milliseconds, and about 600 milliseconds. At 20 milliseconds, she sees this EPR signal, so there's a radical formed. It grows in a little more by 54 milliseconds, and by 600 milliseconds, it's replaced by something else that we know is irrelevant. This is the semiquinone of the reducing system. And so this looks like a kinetically competent radical that forms rapidly upon mixing with the reductant, but then decays. So it's a radical species, and we're all excited. Maria then does some isotopic labeling. Actually, Wei Chen made the molecule with the C2 position labeled by deuterium, and what Maria saw by EPR is a pronounced narrowing of that EPR spectrum, so it's showing relatively strong hyperfine coupling to the C2 hydrogen. So it's substrate derived, and it's got a radical really near that C2 position, maybe on it. And so Wei Chen also made for Maria this thio decanal analog with deuterium there, and the narrowing by deuterium is less pronounced, indicative of less spin density on that carbon when you put the thia group there. And the reason we put that there is that absorbs some of the spin density. So it's consistent with the expectation of a radical at that position or that position. Okay? She also saw there was no change in substituting the enzyme with iron 57, which has a nuclear spin of a half, so it's not coupled to the diironosate. And there's no change in the EPR spectrum when we use C1, C13 labeled substrate, meaning C1 is gone. So this is some sort of product radical. So what is it? Not that way, this one. Well, it turns out Maria looked, and she found in the literature that peroxyl radicals look precisely like this. What's a peroxyl radical? What that is the product of the primary methylene radical produced by ADO in that step in the mechanism, which is then trapped, adducted by a second molecule of oxygen. Okay? So this is not on pathway. This is actually an off pathway product trapped radical. Okay? So Maria got evidence for that by DFT calculations. She can very accurately reproduce the G values of these things and the hyperfine coupling, both for the regular decanal-derived peroxyl radical and for the thiodecanal-derived peroxyl radical. And so her evidence is completely consistent. She then did high-score experiments to directly measure the deuterium couplings here and here, and they are consistent with prediction from DFT calculations. So we know this thing is basically a peroxyl radical that forms when oxygen adds to the initial product that's on pathway. So this thing is an off-pathway product. Okay. So we had two inefficiencies now, and maybe I should walk back to the mechanism. Thought I had an extra mechanism in it. So what we have is, at the point of electron delivery, we think we have a sloppiness when we use this chemical reductant because it puts in a second electron too soon, right? That we could potentially solve by going to the native obligatory one electron donor, ferredoxin, which is what is the donor in vivo. The second problem is when the alkyl radical is generated, it seems that the trapping of that by hydrogen donation is not very efficient. So that if we have oxygen present, it can basically trap that. And of course, that's a disaster. Both are disasters. This step, if you reduce this once and then again, you've used 4 reducing equivalents, 2 here, 1 there, and 1 there, and you didn't make any product. So you're wasting a lot of resources, right? If you get all the way to cleaving the substrate, but then you trap that with oxygen, you don't get the alkane. So you don't get the fuel. So that's even worse. Now you use 4 electrons and you consume the substrate, and you're not getting anywhere, right? So these are two inefficiencies that have come out of this detailed mechanistic dissection. And so we asked the question, well, what happens if we use the ferredoxin, right? So Lauren Rajakovic in the group now did these sequential mixing experiments where the reductant now is actually the ferredoxin. So here she compares the previous experiment, which I already showed, where that peroxyl intermediate is reduced with the chemically reduced phenazine, and these are the kinetics. And she compares that to reduction with the chemically reduced ferredoxin. And what you can see is the ferredoxin is blazingly fast. We barely catch it. In 10 milliseconds, it's almost over. So it really reduces that peroxyl intermediate very rapidly, as you might expect for the natural reducing system. When Lauren looks now at in the EPR, in these freeze-quench EPR experiments, here's the radical that we saw. It was 6%, so that wasn't too bad a deal with the chemical reductant phenazine. With ferredoxin, it's all the way up at 20%. So we used the native reductant, ferredoxin, but now we got a ton more of this off-pathway radical, more than 3 times as much. And so what that says is when we rapidly reduce the peroxo, we get the primary methylene radical, but we then build up more and trap it more with oxygen. And so, the ferredoxin by itself, okay, now I got to figure out how to use a PC. Slideshow. Where's the go-to thing? From current slide, no?
Yeah, I think that should do it.
But the current slide is number 1. No, there we go. All right. So what Lauren reasoned was perhaps the reason we're seeing so much of this trapped radical is that there's so much oxygen. So she went and looked and said, what happens if in that first mix we provide just enough oxygen to make the intermediate and then mix with the ferredoxin? And what you can see is shown here. With O2 saturating conditions, meaning excess oxygen, we get a ton of this off-pathway radical. With O2 limiting, we get a totally different radical species and much less of it. It turns out that's an amino acid radical in the enzyme active site. It's a cysteine radical. And so we basically avoid production of this off-pathway radical by limiting oxygen. Okay? Now that may not be the most useful strategy in an oxygenic phototroph, but we don't really know what the oxygen concentration is in these cultures that Jewel is using to make and certainly LS9's process in E. coli, one could consider limiting oxygen. So what Lauren then did was to look at the relative yield of formate. Now recall, when this radical gets trapped by oxygen, formate has already been made. So the implication of the trapping of the radical is you get formate, you consume the substrate, but you don't get the alkane. And so Lauren looked at the yield of alkane relative to the yield of formate under saturating oxygen, and what she sees is they're drastically different. And this has not been published. Every account of this reaction has shown a one-to-one stoichiometry between oxygen and formate, but those are typically under culture conditions. So this may not be, not culture conditions, but more like air-saturated conditions. So this may be only specific to saturating O2, but what you can see here is that the slope of alkane produced per NADPH, which could theoretically be 1, is down at a pathetic 0.17, and the yield of formate is much better. However, when she goes to limiting oxygen, you can see here with the ferredoxin, now this is with ferredoxin reductase as well, the slope of both under ideal conditions, she now has optimized at about 0.82. And so by use of the natural reducing system and by limiting oxygen in vitro, she can see very close to the theoretical value of unity for the stoichiometry. Okay. So what this tells us is that the enzyme, the unique chemistry of the enzyme presents it with 2 challenges. The first is to handle the delivery of electrons in flight during catalysis in an orderly fashion with proper timing to ensure that one goes in to promote decay of the intermediate, but then a second doesn't go in until after the fragmentation has occurred. And the second challenge is the enzyme, although there are a number of enzymes that can deal with alkyl radicals. In fact, we just published on one in the carbapenem biosynthetic pathway in Science this year, where there's a tyrosine donor to efficiently trap an alkyl radical. Although there are solutions to that have evolved in other enzymes, this guy doesn't really have it. It's just absolutely not very good at dealing with that alkyl radical. And what we conclude is, by mutagenesis experiments, is there is no dedicated hydrogen donor evolved into this enzyme, which is very surprising because alkyl radicals are extremely reactive and could be damaging. And the Marsh group published on the basis of solvent deuterium kinetic isotope effects their belief that the hydrogen donor is actually a bound solvent to the cofactor. That's a weird reaction, and if you present that to guys like Jim Mayer and other really good inorganic chemists who are interested in this stuff, they tell you that's backwards, that wouldn't happen, okay? So we don't really know how the enzyme is dealing with this alkyl radical, but we do know from our experiments it doesn't do it very well. So here's a potential place to evolve the thing to be able to handle that better, possibly do more turnovers, and possibly waste less in cleaving the substrate without getting the alkane fuel out. Okay, so the second place that has been identified by the Shanklin group as a challenge for this enzyme is dealing with reactive oxygen species. So you need oxygen, it's a substrate, the pathway's in an oxygenic phototroph, it's going to face oxygen, and it's going to face reduced byproducts of oxygen metabolism. And so what the Shanklin group showed is that the enzyme's potently inhibited by hydrogen peroxide. But there were enough weird observations in that paper that we wanted to actually go in and look at the mechanism of peroxide inhibition. So these are very preliminary data, but we found some very surprising things. So when Maria looks at the reaction of the reduced enzyme with peroxide, it turns out to be 1,000-fold faster than the reaction of the reduced enzyme with O2. So here's the O2 reaction. You can see the units here are K observed 10 to the minus 3 per second. Here it's K observed in per second. And you can see these traces are on the maybe 500 millisecond time scale. These are out here around 500 seconds. So it's very much faster to react with peroxide than with O2. That is absolutely backwards for members of this family. We have a ton of these ferritin-like diiron proteins in our lab. We've looked at reactions of some of them with oxygen. Here's the ribonucleotide reductase. The second order rate constant is 2.1 times 10 to the 5th. For oxygen, for peroxide, it's 4-fold less. This AURF enzyme, 66 times 10 to the 3rd per molar per second. With oxygen, 3-fold less with peroxide. They all react less rapidly with peroxide, and ADO is 1,000-fold more rapidly. What's also true is, whereas only the aldehyde triggers the reaction with oxygen, the reaction with peroxide is actually triggered also by the alcohol, okay? So that tells you the alcohol binds and does whatever conformational change to that cofactor is required to get it more reactive with peroxide. And so you can see here, by the time you go to the presence of the alcohol, this has been enhanced 2.5-fold relative to the absence of any substrate. And the O2 reaction doesn't change, so now we're talking about almost 3,000-fold different, faster with peroxide. So this is extraordinarily efficient to react with peroxide, and that may be a central challenge the enzyme faces. What's more, ADO itself, in the absence of a triggering compound, either aldehyde or alcohol, and all the other ferritin-like diiron carboxylate enzymes we've examined, react with peroxide smoothly without accumulation of intermediates to go from diferrous to diferric. That's expected. It's electron balance. You don't need any intermediates. Somehow, for whatever reason, ADO, when the alcohol or aldehyde is present and it reacts with peroxide, it makes some intermediate with a much larger delta EQ, shown in red. So I'll show you the evolution. So here's the reaction of the anaerobic protein, isolated protein. So it's diferrous with alcohol bound. And immediately upon mixing with peroxide, in the first 10 milliseconds, we see development of this new quadrupole doublet. It develops to maximum extent by about a couple hundred milliseconds, and then it begins to decay, and we see development of the blue features here with a much narrower quadrupole doublet. So if you compare this one and this one, those peaks are not the same. They're fairly well resolved in the mock spec. It's a little hard to see here, but you can see it when they're compared in this intermediate state. So what we have is rapid development of some intermediate complex upon reaction with peroxide, and then the very slow decay. And what is that decay? We had no idea, but we theorized that because this reaction is unique in not needing delivery of protons during the actual cleavage event, that this might involve a very slow proton delivery event. So Maria, she just did these essentially 2 days ago. She looked at the dependence of the decay phase on the pH and on the solvent isotope, so using deuterium. And what she finds is both a pH effect, it's faster at low pH, and a solvent isotope effect, it's slower in deuterium. And so what this indicates is that the change in state from the intermediate to the product involves some proton uptake. So we think this has to do with the need of the enzyme to basically not protonate the peroxide so it can be nucleophilic, and then not protonate it after reduction so it can do this beta fission reaction. And only after the hydrogen has been delivered to the alkane do we now allow for proton uptake. So that the enzyme is effectively a good proton screen and prevents and causes accumulation of intermediates with in the reaction with hydrogen peroxide. So obviously that doesn't tell us how to fix the peroxide inhibition problem. John Shanklin fused it to catalase. That partially solved the problem, but we think there's more engineering to do here to deal with reactive oxygen species in this enzyme. So I think I acknowledged all the people who did the work. Doug Worry and Squire Booker's group got us started in this area. This has been an absolutely equal partnership among 3 PIs at Penn State. Doug with Squire got us started. Ning took the ball and ran with it to show formate as the product and the oxygenative nature. Maria then characterized the intermediate. Lawrence looking at reduction by the ferredoxin, and Wei Chen has provided substrates throughout. And of course, Karsten Krebs and I run a joint group, so he co-advises all these people except Doug, who's advising Squire. And he's been an equal partner in this, and these guys funded it. And I thank you for your attention.
Hey, I'm sorry if I missed it, but I was wondering why you had to use the methoxyphenazine methysulfate versus just a normal PMS?
You're testing my memory. Yeah, it's because of the photochemistry behavior of one versus the other.
Does the methoxy PMS not photodegrade in its oxidized form?
It still does, but not as badly. So that's why we use it. That's exactly right. But the regular PMS worked pretty well too. So it wasn't like we couldn't have done it. That was just a tactical choice.
What's the function of the enzyme in the original?
Oh, I love that question. I look at it, I'm not a bacteriologist at all, but I look at it as probably the equivalent of microbial exercise. I think it's probably an energy wasting mechanism. You think about how much energy goes into synthesis of a fatty acid, it's a ton. If you think about, cyanobacteria, they may not they're usually probably not replete with light, with infinite energy, but they sometimes find themselves under those conditions, right? So if you don't need energy, and if, sometimes dealing with high light can be a problem in terms of generation of excess reducing equivalents and reactive oxygen species, it may be this is a way to burn 4 reducing equivalents, take an abundant compound and just basically waste it. To my knowledge, they don't care about alkanes. They don't burn them. They don't, So I think it's really just wasted energy, as far as I know. So I have no better answer than to say it looks like an absolute, like you and me running on a treadmill. That's what it looks like to me. So I don't know.
All right, if you have further questions, please discuss with Marty Bollinger over lunch.